Okay, so today I'm going to talk about the brain-heart axis, and I'm going to just give you a brief introduction on the embodied brain and interoception, and then I'm going to present a multimodal study, MEG and EEG, of the heart-brain axis, and then I'll finish up with outstanding questions and the future. Okay, so when we look at the 21st century now and we look back at last century, the priorities back then were that we've really focused on the grey matter of the brain, and we spent a lot of time studying the function of discrete brain regions and where they were located, and generally we spent a lot of time on functional localization. And we did that with EEG and intracranial EEG and MEG as well. And then we also focused on studying inner mental life with respect to what's going on around us, so exteroception. When we look at our 21st century, though, things have changed. And now we focus our attention on connectivity. We also are more focused on studying interoception. And by that, I mean the world inside us, the world within. And this consists of our soft tissues, our viscera, and these various organs send signals to the brain via the vagus nerve, and the brain in turn sends signals to them. So there's a two-way information exchange between these internal organs and our brain. And we've known about this for quite a long time, and people have recorded visceral evoked responses, and in the form of many types. We can record activity from the heart, either electrically or magnetically. We can record activity from the gut, again, electric and magnetic activity. And there have been studies in pregnant women with recording contractions of the uterus as they go into labor, and this has been done electrophysiologically. People have also performed studies on various parts of the digestive tract, the esophagus, for instance, all the way to the rectum, and people have also studied the urethra. And these have been mainly electrophysiological studies in the clinic, in clinical disorders. So we've been focused on visceral evoked responses for quite some time, but in a sort of at a sort of a low level. But I want to talk a little bit more about brain and visceral interactions today. So, without further ado, in the lab, we had the objective of trying to study the dynamic behavior of brain network connections as a function of the cardiac cycle. And so we specifically were interested in knowing what effect the activity of the heart has on different brain rhythms and also on brain network connections themselves. Now, before I go on, I need to just talk a little bit about activity of the heart because everyone mightn't be familiar with it. The heart has two chambers, the smaller chambers, the upper chambers or atria, and the lower chambers known as ventricles. And the electrical activity of the heart consists of a series of waves, which have letters of the alphabet as their names. The P wave corresponds to the depolarization and the contraction of the atria. And this QRS complex here corresponds to the depolarization and contraction of the ventricles. And then the subsequent T wave here, that's the repolarization and relaxation of the ventricles. And so we're going to need this signal. I'm going to talk about this a little bit more in a minute. Our data acquisition This is a multimodal study of the resting state. We recorded 40 minutes of activity. We had 4 subjects, and these are their sex and age characteristics. And we recorded MEG, EEG, EOG, of course, ECG, at a high sampling frequency. And we also recorded whole head, high-res anatomical MRI scans in all the subjects. And this was work done with my lab and also collaborators such as Olaf Sporns at Indiana University, and then folks with at the Paris Brain Institute in the lab of Nathalie Georges, Laurent Hugueville, and Christophe Guiton. Oh, and I forgot to mention, Kami Salibaeva has done all of the analysis for this study, and she successfully completed her PhD in my lab in May, and she's currently doing her postdoc at the University of Newcastle in Australia. All right, so let's talk a little bit about data segmentation, because this is very critical in this study. We first of all pre-processed the data to remove EOG and ECG and EMG, and then we took the artifact-free data, and now we epoched them as a function of the cardiac cycle. And so that meant taking very, very brief segments of MEG. So here's our ECG waveform again with the labeled waves. And so what we did was we took a time interval from the peak of the R wave to the end of the T wave here. And that was our systolic period. And then we took another period following the end of the T wave, and we called that our diastolic period, and that's where nothing is occurring in terms of the activity of the heart. We made sure that those two epochs are equal, and we looked at customizing these intervals in each subject, and I'll tell you about that in a minute, but these were made equal. We, for each subject, first generated a systolic duration distribution, because everybody's different, and also different heartbeats are different. And so this is for subject 1, over 2,200 heartbeats. This is the distribution of the systolic duration distribution. And then we selected the minimum duration for epoching data for each subject. And in this case, we selected 350 milliseconds, as I showed you on the previous slide. And so, of course, this has, this period was different for each subject. They were fairly close together, 350, 320 milliseconds, so somewhere in that range. But of course, this has implications for the investigation, because we can't study some of the slow EEG rhythms, MEG rhythms. Okay, so once we'd chosen our segmentation intervals, we took our signals and did the standard kind of pipeline for functional connectivity and network analysis as a function of canonical frequency band. And we filtered the activity in each of the 5 ranges. We source localized the activity, and then we averaged the sources to parcels within the brain. We used the Distru Atlas for our parcellation. And then we took for each of these parcels a time course for each parcel. And in our case, we used power envelope correlations. And then once you've done that, you can estimate a set of, for all of the parcels, you can generate a set of phase or amplitude couplings. And of course, we had power envelope correlations, as I mentioned. And this figure is adapted from this really lovely didactic paper by Sadayani and Wusic. So our data analysis, just to remind us, the digital sensor locations were all merged to our MRI scans, source localization, and here's just a reminder of the parcellation scheme that's through Atlas, just showing a lateral view. Now, data analysis, a fairly complex slide here, but basically I've already explained, we epoch the data by diastole and systole for each subject and each frequency band. And then we had to generate a null distribution for comparison. So what we did was we took these data and we shuffled the labels 1,000 times. And we can talk about the null later, but that's how we chose to do it. We did power envelope correlations in each of these data sets. There are about 10,000 of these. And here we're showing just a difference calculation of the matrix here, which just for display. We took the power envelope correlations across systole and diastole and compared them. There are about 10,000 calculations, about over 10,000 calculations. About 10,000 of them were non-significant, 147 were significant, and they're the ones I'm going to talk about in a moment. This is just the results of our multi-resolution consensus clustering, looking at community behavior across diastole and systole. And here we've got Olaf Sporns had reordered systole here in the same sort of order as diastole. So you can see there really is a difference across the cardiac cycle in brain network behavior, which we'll look at in a minute. Okay, so I see there's a question. No, there's not. Okay. All right, so let's talk about these 147 functional connectivity linkages that we saw. Just for your memory, this is just a look at the Stru Atlas, the views of the parcels, and we can then draw links between them. And what I'm showing you here on this complicated table is we're looking at gamma band results across all four participants. And what we've got here is the results of the 4 participants with each functional link shown here. And to make it easier for you, because you don't know these links by heart, and neither do I always have to reactivate my memory. These numbers here are in these lobes, and so that will make it a bit easier for you to see. And just see how this works with the stuff that I've highlighted here. Their area 60, for instance, is shown here. Its neighbor is 21, and there are neighboring areas 11 and 44 that have these connections. And so that's just one example of some of the connections that are being made here. What's interesting about gamma activity is it's can be within the same lobe, or it can be within neighboring lobes. And the other thing about it is that you can have either inter or intra-hemispheric connections, right to right and left to right, for instance. So that's gamma activity. I'm going to do the same thing and look at beta and alpha. Beta was a bit more complicated. When we looked for across all 4 subjects, there were no significant functional linkages. So we took them, the subjects in triplets, and that's what I'm showing here. And the table's arranged in exactly the same way as before. And so now we see something different in the beta activity. Now we start to see longer range connections, occipital frontal, frontoparietal connections. So that was an interesting feature of beta activity, which we didn't see in gamma, because that, remember, had within lobe connections. And we also had here neighboring lobe connections as well, and both intra and interhemispheric connections. Now let's turn to the alpha band. No significant connections across the 4 subjects, and across 3 subjects, we found quite a number of links. Again, now alpha shows a slightly different pattern. What we see now is we see within lobe connections, and we see across neighboring lobe connections, but typically these are all within the same hemisphere. Very different pattern to what we saw with gamma and beta. Network architecture and community structure. This is the results of our multi-resolution consensus clustering analysis. We've defined communities as greater than 10 nodes, and we have what we call a core and periphery structure. In our case, the core comprises about 80% of all the nodes, and they're typically interconnected, and that tends to be comparable across subjects. And then our periphery consists of sparsely interconnected nodes and lone nodes. And so what I've displayed here are the subjects, the bands across systole and diastole, and of course this is impossible to look at with numbers, so I'm going to show you some diagrams, and hopefully that'll make a bit more sense. And let's look at alpha activity and what happens across the cardiac cycle. Nodes can either stay and remain stable across the cardiac cycle, or they can move. And so the majority of nodes are stable across the cardiac cycle, and they're common across the 3 subjects. And here they are. And then across the different subjects, there are different, slightly different patterns of movement. So that's the behavior for alpha, better seen than that unwieldy table I just showed you. And let's have a look at beta in the same way. And remember, now we've got 4 subjects. Just to make it a bit easier for you, this is the total number of nodes in each of these categorizations. Again, in the beta range, like alpha, lots of stability across the cardiac cycle, commonality across all 4 subjects, and then, of course, there are these various other intersections across the subjects. And not so much movement, similar to alpha. Let's look at gamma now. Stable and moving. Now we've got a slightly different arrangement. These are the total number of nodes again across 4 subjects. And now we've got a lot more kind of equality. We've got movement across the cardiac cycle and not quite as much stability. And so that's the gamma band behaves differently to the alpha and beta. Let's look at now across node movement and within lobes across the cardiac cycle. Let me explain this complex Sankey plot. It's not that complex when we look at it close up. We're displaying the Strew Atlas parcels, and they're moving. They're either static or they're moving, and they can be. The core will be the one that will be seen here, as you can see that stays. There's no movement across the community. This is the movement here is just for display purposes. The color coding are the lobes here. We're showing 3 subjects in the alpha range, and you can see that the core, it's the largest part of this plot. Here's the periphery, and nothing's changing. Moving nodes across the cardiac cycle. There are a few of those, and you can see those vary, and you can see the differences across subjects here. We've got 3 of the 4 subjects shown here in the alpha range. Okay, same thing for now beta and gamma. Again, a large stable core, small periphery, but different across subjects. And then here we've got the nodes that move across subjects. And then as we go to gamma, there's more movement. And this is just looking at the data in a slightly different way. So hopefully that makes sense, because it's a fairly complex data set. Mutual information is something else that we calculated, just to try and see whether we really are getting something real, I suppose. We calculated mutual information for each subject's data set relative to their own null. And then rather than showing you these, trying to decipher these numbers, I've plotted it here on the screen, showing you across the 3 frequency ranges, the mutual information for the real data and the corresponding shuffled null. And so there really is something going on, in our opinion, during the cardiac cycle, particularly in the gamma range, which gels with the other results that I just showed you. So interpretation and implications. Well, there are effects across the canonical frequency bands for the MEEG data. The functional linkage length in the beta range, those functional connectivity linkages appear to be longer. They appear to be shorter in the gamma. That was where we had the within lobe ones. This is where we had the fronto-occipital and frontoparietal connections. And then the alpha band was intermediate. The hemispheric pattern tended to be ipsilateral in the alpha range. And then in the beta and gamma range, it was ipsilateral and contralateral. With respect to the core periphery connection pattern, the nodes could either stay or move across communities during the cardiac cycle. And for the alpha and beta range, the majority of nodes stayed static, whereas the gamma was the one that tended to show more movement across the cardiac cycle. I tried to show you that there were individual differences in the functional connectivity changes across the cardiac cycle. And what I couldn't show you so well, but when you actually bury yourself in the data, is that there are actually insular functional connectivity linkages with parietal, limbic, and temporal lobes as well. And of course, the insular is a well-known kind of center for interoception. So we have a lot of limitations in this study, and let me share some of those with you. I already alluded to one of those earlier, and that's the heart rate interval duration. It's not possible for us to do theta and delta band calculations because of the subject's heart rate. And our subjects didn't have high heart rates. I mean, they were 55, 60 beats a minute, maybe a bit over 60. So they weren't high. But even with these low heart rates, these calculations are not possible. We have limited null distribution options. We chose to swap the labels of our data. And we can't switch the heart off during resting state. So that's our big limitation there. And then, of course, I showed you the Strew Atlas parcels, bordering parcels that are coming up and showing us these functional significant linkages. Well, we're going to have inaccuracies with those border parcels. If you've got, the main activity of the source sitting kind of on the fence between the border of those parcels, it could fall either side of the fence. So we're going to have that, and we have to take that into account. And then, of course, there are no subcortical structures included in this analysis, and our N of for the sample was small. So turning towards the end of things, outstanding questions. For me, there are lots of them. This is only a subset. I want to know how these functional connectivity linkages change across the cardiac cycle when our states of consciousness change. I mean, how does it change during sleep? What about when we're walking around? And what about if we have various cardiac or cardiovascular abnormalities, and if we have a neurological disorder. In particular, we've known about the link with the vagus nerve and epileptic seizures for decades. So there's a lot more to unpack there. I already mentioned the fact that we didn't look at subcortical structures. Well, what about the cerebellum? Cerebellum has far more neurons than cerebral cortex. So what happens there during the cardiac cycle? And then what about heart rate variability? With respiration, the beat-to-beat intervals change. How does that affect brain functional connectivity? And then I didn't even go there initially, but the autonomic nervous system. We have parasympathetic and sympathetic systems that are part of that. And we can assess the activity of those, let's say, pupil dilation or neurohormonal assays. We haven't tackled that at all. So in conclusion, seeing the heart-brain axis at work, we see it in the form of altered brain functional connectivity. And so I would advocate that we need to sample bodily signals in addition to brain activity when we're doing these studies. And at a minimum, we need to be recording ECG, I would say. And I think we need to start considering both exteroception and interoception. We forget about the world within. And we should think about this during both resting state and task-evoked studies. And then finally, I would like to acknowledge our sources of funding. And this is these data were collected in a joint binational grant, a cooperative research in the computational neuroscience grant that I hold with Nathalie Georges in France, which I'm very, very grateful for. So with that, I thank you for your attention.